Hello, in this podcast we're going to be learning about mutations and their possible effects. Here we have sickle cell anemia, which we've discussed in the past. On the left here we have normal red blood cells, and here you see that they're all circular shaped. But here on the right you see we have a mixture, and if you see there's one sickle cell shaped. A sickle cell looks a bit like a banana. And this disease is common among sub-Saharan Africans and slightly less common among African Americans. Now here's why it's a problem. The sickling of the blood cells occurs when oxygen levels are low. This could occur frequently, like say if you're exercising or something like that. And what it can do is that the sickle shape can block the flow of the blood, and that can cause pain and fatigue. Now, why do these red blood cells sickle? If you look over here, we have pictures of the two proteins. We have normal hemoglobin and the hemoglobin with the sickling allele. Normal hemoglobin won't clump together, but the shape of the sickling hemoglobin is such that it can clump together. So it's the clumping together that causes the sickle shape, and that's of course a bad thing. But there is a good thing. If you're living in sub-Saharan Africa, which is why it became so common, is that when an individual is heterozygous, they don't have enough of the sickling allele to cause it to clump up so much to make the sickle shape, but the conditions in the red blood cell are such that the malarial parasite, or the parasite that causes malaria, will be killed. So someone who has this gene, if they're heterozygous, they will not come down with malaria, and someone who has normal hemoglobin will. So for that reason, it evolved to become common in sub-Saharan Africa. It's less common among African Americans because malaria has basically been wiped out since, I think, the 1930s and 1940s. So now it's in the United States, it's just negative, while in Africa, it's common. It's more common. And this, by the way, is something to keep in mind about mutations. Mutations in certain circumstances can be beneficial, can be good for you. In other circumstances, they can be bad. All depends on the environment. Why does the protein make a different shape? If you look here at the DNA, we'll notice that there's just a change in one base. An adenine turns into a thymine. Just one base change. That one base change causes one change in amino acid, where glutamine is changed into valine. That one little change causes that change in the shape that allows the clumping and makes a person have sickle cell anemia if they're homozygous and resistance to malaria if they're heterozygous. Now, what is a mutation? We've sort of already said it. It's just simply a change in the DNA. Now, here are the potential effects of a mutation. The mutation can be negative, and an example of that would be Tay-Sachs disease. We've discussed this before. That's where the fat deposits, I think, can't be removed in the brain, and the baby basically, instead of once reached like 6 months, they stop making developmental advances and start going backwards, and they're all dead by the age of 5. Negative mutations tend to be rare. Mutations can be positive. Like one example would be lactose tolerance, not intolerance. Thousands of years ago, all humans were lactose intolerant, just like practically all mammals are lactose intolerant. Lactose intolerant basically means that once you're past the age of infancy, once you stop breastfeeding, you can no longer produce the enzyme that makes milk. And normally this makes perfect sense. Why make an enzyme when milk is not part of your diet? But at some point there was a mutation, and that mutation allowed certain populations to be able to digest lactose, and that expanded what we could eat. Since we were raising animals that produce milk, we were able to add milk into our diet and other dairy products. So that was a beneficial mutation. And there are people who have it, and there are some populations where the lactose tolerance gene lactose tolerance allele has not really entered it yet. Like I think among Aborigines in Australia, I think something like 90% of them are lactose intolerant. Another way that mutations can be positive, and this is used in agriculture, it's something called mutation breeding, where there are certain things that can cause mutations. We'll get into more detail later, and among that would be radiation, like X-rays. And they expose seeds to radiation, The good mutants, the ones with the beneficial mutations, are then bred, and then they're used for food products or other things. And an example of that would be pink grapefruits. Prior to pink grapefruits were either had white. The fruit part was on the inside was either white or red, and actually red was also a mutation too, which occurred in I think 1929. Before that, there were no red grapefruits, and these mutations are also rare. The one reason why these two are very rare is that very, very little of our DNA has genes. Most of it is junk, so most mutations are actually neutral. And another way a mutation, by the way, can also be neutral would be there can be a change in the DNA which does not change the amino acid at all, because you could have several different codons that code for the same amino acid. So a change in one letter could end up with the same amino acid. And we saw this in a lab that we did recently, comparing the gene for cow insulin to human insulin. And as I said, these neutral mutations are common. One thing you really have to remember is this last line here, and make sure that's in your notes, is that in order for a mutation to be heritable, it must occur in a gamete. If it occurs elsewhere in your body, it can cause good things or bad things to the person's body, but unless it's in a sperm or an egg, there's no way it can get into the offspring. Now we come to different types of mutations. First one, which we already hit in looking at karyotyping, is chromosomal mutations. Here we have an example of a karyotype. Karyotype is where you're just simply looking at the chromosomes themselves. If you look at down here, chromosome number 21, you see the 3 copies of it instead of 2 like for everything else. And so this individual has a disease called trisomy 21, also known as Down syndrome. This is caused by an error in meiosis. This type of mutation is more common among older women than younger women, simply because the eggs are much older and more prone to error. In most cases, I would think with chromosomal mutation, the mother doesn't even know that she was pregnant. The effect is just so drastic that it just ends in a miscarriage. In most cases, the woman probably would not even miss her period. Now, point mutation is sickle cell anemia is an example of that, and there one base is substituted for another. Deletion mutation is where one or more bases are removed from a gene. If it's not in multiples of 3, then you end up with a frame shift. Remember, a codon is 3 bases. So if you get a frame shift where everything gets moved over by 1, then every single codon is going to be different after that mutation, because the frame is shifted, so that you'll end up with a very, very major change to the protein, and probably in most cases the protein will stop working. Cystic fibrosis is an example of that, and this was an example. The reason why this is not as major a change, where the protein is still made and still to a certain extent works, is that it's not a frame shift mutation. 3 bases are deleted, so that removed 1 amino acid, phenylalanine, and a person who has this has thick, sticky mucus, and that causes frequent lung infections. Someone who has this disease, they live a short life. We've managed to expand the life quite a bit more, that now they're able to live 37 years, while before it was 6 months. And an example of a deletion mutation which actually is beneficial is that you have 32 bases deleted from a gene called CCR5. A person who's homozygous in that is resistant to HIV. That person will never get HIV. If a person's heterozygous, then they could have HIV, and getting AIDS will be significantly delayed. Also, you have addition mutations, and for this, it's the opposite of a deletion mutation, where instead of bases being deleted or removed, they are added. A gene can also be duplicated. An advantage to that, if the population has 2 copies of a gene, then one gene is free to mutate and turn into a whole new gene. You can get frame shifts with this if it's not multiples of 3. Huntington's disease is an example of an addition mutation. Now here we have cancer, and I put cancer in here because a lot of cancers are caused by mutations. Cancer is basically uncontrolled cell growth, where a cell will just uncontrollably grow and will basically crush the organs in the body, because there's nothing to stop it from growing. They're also able to live throughout the body, where that's where the cancer spreads. Remember in the book we're reading on Henrietta Lacks, the cancer was spread throughout her whole body. There are many mutations that can cause this, and they can affect regulatory DNA, they can affect genes themselves, or they can affect the parts of the DNA that control the genes. And about 90 to 95% of all cancers are caused by the environment. The remainder are genetic. Now, since it's caused by the environment, that means there's actually something you can do about it. You can get the things that cause it out of your environment. These things that cause the mutations are called mutagens. And these mutagens can cause cancer. Now, these are what mutagens are. It's an environmental factor that can cause mutations. Examples of mutagens could be radiation. You got ultraviolet radiation. That's what can cause skin cancer. X-rays and gamma rays. Everyone knows with X-rays now, your dentist or dental hygienist always leaves the room when they take an X-ray, and that's because they could be severely overexposed to the X-rays, and that can cause problems. Originally, when X-rays first came about, people were using them in shoe stores to fit your shoes. This is before they knew that X-rays can cause cancer. And it caused serious problems in the shoe salesmen because they always demonstrated it with their own feet, where you put the shoe on, put in the X-ray machine, and you would look at how the shoe fit using the X-ray machine. And a lot of these retired shoe salesmen end up with lots of cancers in their feet. Nicotine, which is found in tobacco, is a mutagen. And so if you're smoking and you stop smoking, we've also passed laws where smoking is not allowed in many areas where it used to be allowed, like in restaurants and airports and airplanes. That basically has probably saved many, many lives. Also, something called free radicals cause mutations, and this forms normally during metabolism. It can also be more common in certain types of foods, like if you're barbecuing and you really overcook the food, that is high in free radicals. It's also found in herbicides, so it's very important for you to really wash your fruits and vegetables before you eat them. And it's also found in nicotine. And what you can do about it is the antioxidants destroy them. So have lots of vitamin C and vitamin E in your diet. Basically, eat your vegetables. Now we come to our concluding questions. Number 1, what is a mutation? Number 2, what kind of mutation causes the sickle cell allele? Number 3, when is a mutation heritable? Number 4, why is cancer included in this podcast? And number 5, list 2 examples of mutagens. So that concludes this podcast, and I'll see you in class tomorrow.